and start. Thank you.
Yes, thank you very much for introducing me, Chris Sensei, and hello again for DTMH students, and also hello for TTM students. It's been since April self-introduction sessions, and as Chris Sensei introduced me, I'm a student in DTMH, so you may think it is very strange thing to give such a talk, such a student make such a talk to other students. Vaccine is one of my special interests, and I've been made more than 20 times lectures regarding COVID vaccine in these 4 months in Japan, and it's kind of hobby for me to talk about COVID vaccine. So I don't think this is a lecture from me, but this is just a talk or a discussion. So please be relaxed and sit back, and you may drink something. Of course, I shall not drink, but please feel free to discuss about COVID vaccine. And during my lecture, please do not hesitate to stop me and ask some questions or add some comments or correct my errata. I very welcome interaction with you. So I'll start my presentation. And before starting my presentation, I put a URL on the chat box to download my handouts. Let's see. I put the link for download handout PDF for my lecture slides. Then now share my screen. Yep. Okay, here we go. So today's title is Current Overview of COVID Vaccines as of 10th of June, as of today. And my background is, as Chris Sensei mentioned, I've been family physician. I had been family physician for 18 years in Japan. And during my during my career, in the latter half of career, I got special interest to vaccines and travel medicine. And I switched my job to quarantine officers 4 years ago. And now I'm a student, and I'm planning something after finishing this DTMH course. Actually, I'm planning to establish a small company to provide education of vaccines and travel medicine for Japanese healthcare professionals. And it's still just in my mind, and I have not done anything for my plan, but I'll do my best, and I appreciate your help or for my future dream. Okay, and these are part of my works during COVID pandemic period. As quarantine officer, I provided some online seminars, or I wrote various kinds of review articles on internet or on medical journals. And I gave a talk about border measures of COVID-19 invited by Japan Clinical Virology Academy, and talked with Professor Minishura, 80% uncle, or Dr. Oshitani, as we learned from him just before about COVID and SARS, and here Kutsuna Sensei. And also I wrote a review article for COVID vaccine in Nihon Eiji Shinpo, and this is a screenshot of a YouTube lecture held by my Academy of Family Physician in Japan Primary Care Association. Now it's still on YouTube, but this was taken in February, and it's already outdated. So I was thinking of necessity of renewing the lecture. And suddenly last night or before, Ministry of Health suddenly contacted to our academy and requested to renew the this video lecture. And they said deadline is 14th Monday, please. Oh, I was surprised. What a crazy end. But that they say they are thinking to provide a renewed video lecture for the newer vaccinators in Japan. They are thinking to involve lab technicians and sorry, I forgot the English name. EMS, emergency medical something, and they think to provide my lecture for them to be a new vaccinator of COVID vaccine. So I make my best effort to provide a new one after this English talk. Yeah. So today's talk is it's such kind of a non-systematic review of medical literatures regarding COVID vaccines. I read around 100 original articles or letters or correspondence relevant to COVID vaccines in these half a year. And every week we are seeing new medical articles of COVID vaccines, 1 to 3 articles every week. So it's very difficult to catch up the new knowledge. And today's my talk is, of course, subject to change in the near future because we are seeing new knowledge every week. So today's my talk would be wrong partly would be wrong in the near future. So please keep your eyes on every new article. And today's my talks consisted of these 6 factors. Okay, and so let's move on to the first one. The first one is classification of COVID vaccines. And I climbed up the Mount Unzen several days ago and found the world's oldest COVAX bottle on the top of mountain. It's just a joke. Okay, here is a schema of virus, and coronavirus looks like this round-shaped enveloped virus. And as you know, SARS-CoV-2 has some surface proteins on the envelope, and one of the proteins named spike protein is the key to embed into human cells. So the target antigen for vaccine is also spike protein. So that developing COVID vaccines also means developing spike, how to produce spike protein as vaccines. So now here we have some kinds of development methods for vaccines. And for COVID vaccines, we have now messenger RNA vaccines, viral vector vaccine, inactivated vaccine, and protein-based vaccine. And as you know, we also have a live attenuated vaccine as a general rule, but so far the world has never been successful to establish live attenuated vaccine for COVID. So now we have 4 types of COVID vaccine. And as you know, messenger RNA vaccines contains a fragment of viral RNA coding spike protein. And this fragment of messenger RNA is wrapped by polyethylene glycol, PEG. This is important potential anaphylactic allergen. I'll talk about it later. So in this messenger RNA vaccine case, if we are shot by this vaccine, our muscle cells get this fragment of our viral RNA, and our muscle cells produce spike protein inside our cytoplasm. So we ourselves are the factory of spike protein. We are the factory of vaccines in case of messenger RNA vaccines. And on the other hand, viral vector vaccines contains a live virus other than coronavirus, other than SARS-CoV-2. And this virus is harmless for human, but has ability to invade human cells. And this harmless virus has, of course, its own gene, and this gene is recombinant, embedded by a spike protein coding fragment of coronavirus. So if we are shot by this vaccine, our human cells were infected, invaded by this harmless virus, and this virus excrete its gene into our cytoplasm or into our nucleus. And then our cells read this part of spike protein coding gene and produce spike protein inside our cytoplasm. Again, our body itself is a factory for spike protein. And I don't think I need to explain details about inactivated vaccines. It's very simple. Culture the virus, culture the SARS-CoV-2, and add formalin, then virus is be destroyed, and purify the debris, then we can get spike protein from the debris. And the last one is protein-based vaccines. This is similar to viral vector vaccines, but this one use genetically recombinant plant virus, a certain virus which invade only to a certain plant is recombinant and embedded of a coronavirus spike protein coding RNA into their gene, and that virus infect to a certain plant cells, and that plant cells produce spike protein and purify those spike protein and to utilize vaccine. This is protein-based vaccine. This is already kind of tradition. It has a history of more than 15 years or 20 years. It is famous for hepatitis B vaccine, or most of us are vaccinated as healthcare professionals. Hepatitis B vaccine utilized in Japan is made by this technique, recombinant protein. So these 4 techniques are used for COVID vaccine so far. And in reality, now we have 10 total. In total, 10 vaccines are utilized worldwide. 2 messenger RNA vaccines, 4 viral vector vaccines, and 4 inactivated vaccines. Most of them are needs twice dosing, and some of them, only 2 of them, 2 viral vector vaccine is only single shot. And Japan has approved only 3 of them, 2 messenger RNA vaccines, Pfizer and Moderna, and 1 viral vector vaccines from Oxford, AstraZeneca. And according to New York Times site, COVID vaccine tracker or something like that, its name, according to the site, as of 3rd June, total more than 2 billion doses. So it means more than 2 billion people have got the shots of COVID vaccines. 2 billions, surprising number. So then now we look into the efficacy or effectiveness of COVID vaccines. Here is a summary table of phase 3 clinical trials. These are shown vaccine efficacy of each vaccine against symptomatic COVID. And most of you may know messenger RNA vaccine showed very, very, very high efficacy against symptomatic COVID, around 95%, and followed by viral vector vaccines. They showed moderate efficacy against symptomatic COVID, around 17. And this one, Sputnik 5, I'm not sure how to pronounce this one, Sputnik 5 or Sputnik V. Recent Sputnik showed more than 19% efficacy against symptomatic COVID. This is very good, but be careful. I'm not sure why, but this article showed the efficacy just before the second shot of their vaccine. I'm not sure the reason. And inactivated vaccines have been developed by China and India, and their efficacy is a bit lower than viral vector vaccines, around 50 to 70 or 78. But most of them are not, they have not released medical articles on peer-reviewed journal, and we can just see the governmental release or pharma press release. So I cannot assess the details of these vaccines. So here we can summarize as this: messenger RNA vaccines has fairly reduced symptomatic COVID, approximately 95%, followed by viral vector vaccine on these figures, and moreover, followed by inactivated vaccines around these figures. They are very effective. They are very efficacious against symptomatic COVID. Symptomatic COVID means, you may know, in the clinical trials, they divide participants in two arms. One is true vaccines, and the other is placebo. Most of phase 3 trials used just saline, just water. And each arms of participants were tracked for a certain period. And some of them had some symptoms like a fever or cough or pneumonia or hospitalized due to something. And all of them were tested by PCR from nasopharyngeal swab. And in the true vaccine arm, very few people showed positive result for COVID. And in the placebo arm, a certain number of patients showed positive against COVID. And the differences between two arms is this figure, 95% or 65%. This is a reduction of symptomatic COVID by these experimental vaccines. Okay? So here we recap the infection of SARS-CoV-2 exposure. Once a human is exposed by SARS-CoV-2, the human can be each of these patterns. One get test negative results, and some of them truly have no infections. but some of them even getting negative test results, but still they may have virus in their body. So it means false negative. And false negative virus infected patients may transmit the virus to other people. So these false negative patients still make onward transmission to other susceptible people. And on the other hand, some SARS-CoV-2 exposed people get a real disease, symptomatic ones, or some people get severe form of COVID. And some people get virus and get positive test results, but still asymptomatic, as you know. And of course, these asymptomatic positive patients still make onward transmission, passing virus to other people. These are happening on COVID. And as we saw the phase 3 trials results, we can say vaccines, COVID vaccines flip this symptomatic positive to negative. Okay, trials showed that vaccines flipped symptomatic positive to negative in the extent of 50 through 94%. This is what vaccine made. So our next interest is, clinical trials showed flipping effect of against symptomatic COVID. Then what is happening on asymptomatic COVID or severe COVID? Do they also flip these patterns of positive to negative? This is the second question. So this should be investigated in real world. And this is again the same table. And from these vaccines, real world data have been unfortunately obtained from only these 3 vaccines. Other vaccines, as much as I know, have not been well investigated in real world. So from now on, we are looking into these 3 vaccines real world data. Okay, then first, we look into messenger RNA vaccines effectiveness. In this table, I summarized 5 independent researches in real world. They are done by Israel or US or UK, and their research design was cohort design. Some of them are historical, some of them are prospective cohort, and utilized vaccines are Pfizer, singly Pfizer, or Pfizer and Moderna. And these are effectiveness against each pattern of disease, symptomatic COVID or hospitalization due to COVID, severe form of COVID, or asymptomatic COVID, and any kind of positive results against COVID test. And as you know, as you can see, they showed almost same effectiveness with phase 3 clinical trials. So now we can summarize messenger RNA vaccines reduce or flip positive to negative in every pattern of COVID to the degree of approximately 90%. Very, very highly effective are messenger RNA vaccines. Next, we look into viral vector vaccines of Oxford. These are, here we have 2 studies. Here we have 3 tables here, but this one and this one is from the same study. And you may know the UK government decided to give Oxford vaccines or messenger RNA vaccines for their national people by the interval of 3 months. Even though Oxford vaccines was developed by divided dose of 4 weeks interval, but UK government decided to prolong the interval in order to distribute more vaccines, more first vaccines for more people. So the real world data from UK shows 2 patterns. One is real world effectiveness after 2 doses, and the other is tentative temporary effectiveness after just single dose. And this table showed the 2-dose effectiveness. And as you may see, symptomatic COVID was reduced by 63.1%, and any form of positive COVID were reduced around 50%. But unfortunately, they couldn't show the effectiveness against asymptomatic COVID. NS stands for not significant. And when we look into the single dose one, they investigated the temporary tentative effectiveness against COVID. And this study investigated hospitalization reduction after single dose. And the effectiveness increased according to the time. So these are days, so it means like in the 2nd week, 3rd week, in 4th week, 5th week, reduction of hospitalization due to COVID increase according to time. And but in the latter part of period after first shot, they couldn't find any differences between vaccinated and unvaccinated because the sample size, outcome patients number was quite low, and they couldn't do statistical analysis. And in this study, they also investigated the reduction of symptomatic or asymptomatic or any form of COVID positive after the first shot. And it also slightly increased according to the time, but after 91 days, after 3 months has passed, they suddenly lose their effectiveness against symptomatic COVID or any COVID positive, not significant. So here we can summarize, single dose of Oxford vaccine keeps its effectiveness up to 3 months at the longest. And from after 4 months, they lose their effectiveness. So we can say we should do definitely the 2nd shot of Oxford vaccines at least by the end of 3 months. And now, as you know, various kinds of variant strains are spreading worldwide. And our interest is also kept on the vaccine effectiveness against each of variants. And this table summarizes their effectiveness. And as you know, recently WHO changed the variant's name into Alpha, Beta, Gamma, Delta. And for Alpha strain, formerly known as UK strains, was investigated by these 2 studies. And Pfizer vaccines fortunately showed almost same effectiveness with the original strain. Here, any COVID positive was reduced by around 95%, for severe form 100%. And in this study, also any positive COVID was reduced by around 90%. And also in this study, Oxford vaccines also showed almost same effectiveness with the original strain, around 70%. And when we look into beta strain, formerly known as South African strain, unfortunately, Oxford vaccine failed to show effectiveness against beta strain. But Pfizer vaccine still showed the high efficacy, high effectiveness. And for Delta strain, formerly known as Indian strain, Pfizer vaccine luckily showed the high effectiveness. Congratulations. Now Japan is being hit by the huge wave of Indian strain. But if we've got shot of Pfizer vaccines, we may be protected even from Delta strain, from Indian strain. And Oxford vaccines also showed a high effectiveness against Indian strain. And unfortunately, I couldn't find any studies done by true endpoint against gamma strain here. And Moderna vaccine is also not investigated for these strains, but theoretically, Moderna vaccine and Pfizer vaccine may have same mechanism against virus. So we can expect that Moderna vaccine may also have same effectiveness against these strains. So this is a summary. Messenger RNA vaccines reduce any COVID like this, and Oxford vaccine also reduce like this. Its effectiveness is a bit lower than messenger RNA vaccines, but definitely higher than influenza vaccine against seasonal influenza. Okay. So here again summary schema. These vaccines flip any kind of positive from to negative. Congratulations. But now what about false positive? Okay. So we should ask next, do the vaccinated people still transmit the SARS-CoV-2 to other people, or do those vaccinated people never transmit the virus to other people? This is not resolved from researches we have seen here. So which is the truth is the next question. But proof of truly no COVID is probatio diabolica, devil's proof, or in Japanese, akuma no shoumen. It is impossible to prove that this patient does not have the virus, definitely. It is because any COVID tests are quite less sensitive. Each COVID test has low sensitivity as high as 70% in case of PCR tests. So each negative test results cannot exclude infection of COVID. That is the reason why impossible. But here we have alternative. We can prove the reduction of onward transmission from vaccinated people to other susceptible people. If we can prove this one, we can say if if we investigate the onward transmission, and if that onward transmission from vaccinated people is reduced, then we can say vaccine did not make such false negative people, but make this truly not infected people. Do you understand what I'm saying? It's a very tricky, but here we go. And it is a bit tricky how to investigate reduction of onward transmission. Here is the schema. Here we have unvaccinated guy here and contacts around the guy. Here we have vaccinated guy and also contacts around this vaccinated guy. And we observe, we track these contacts as well as the vaccinated or unvaccinated guy for a certain period. And in this case, contacts around unvaccinated guy may get some infection like X, number of X. And here, contacts around vaccinated guy also may get infection. Then, but here, the number of infected people matters. If the number of contacts infection is lower than X, then we can say onward transmission from vaccinated guy is lower than onward transmission from unvaccinated guy. Do you understand this a bit tricky schema? And in reality, 2 studies have been done in UK in terms of this onward transmission. And the one study showed such a thing, utilized vaccines or Pfizer or Oxford, and hazard ratio of any COVID in unvaccinated contacts of vaccinated index cases or blah, 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 compared to unvaccinated contacts of unvaccinated index case was 0.46 to 0.50. And the next one, this is also from UK. The previous one was from Scotland, and the next one is from England. It also showed almost same results. Odds ratio was in range of 0.43 to 0.67. So here we can summarize. Pfizer and Oxford vaccines reduce onward transmission in approximately 50%. And here, please ignore the precise conversion of odds ratio or hazard ratio to relative risk. It's very it needs very complicated calculation, complicated formula. And if you are interested in this issue, please you have to refer to Sharon Sensei. I'm not quite sure of this issue. But here we can say Pfizer and Oxford vaccines showed cocooning effect by vaccination. And these results suggest that these vaccines make vaccinees truly free from infection to a certain extent. To a certain extent, it's of course 50% showed by these studies. But be careful, behavior of unvaccinated contacts could be heterogeneous. Some, I forgot to mention the contacts about on these studies. These contacts are household contacts. They are living with unvaccinated guys or vaccinated guys. But these household contacts behave heterogeneously. Some household contacts may have kept in staying home, and the others might have go out for their job, or might have went out for their shopping, and might have went out for pub to drink. So their behavior could have been heterogeneous and could not adjust in these studies. So we might expect that this certain extent might be around as high as 90%, same as reduction of positivity. This is just my expectation, but this is a possibility. So Pfizer and Oxford vaccines showed cocooning effect. Here is the explanation of schema of cocooning effect. Blue guys are not immunized, not vaccinated people, so it means susceptible. And yellow guys are immunized and protected from infections. Red guys are not immunized and because of it get infected. So if many of group blue, there are only many of blue guys, not immunized ones, and put infected guys, two infected guys, then this community, these people easily get the expansion of infections. And if only a part of people get immunized, if we put two vaccinated guys, two infected guys, still this community has a great expansion of infection, and only immunized people were protected. But if majority of people were vaccinated, immunized, and put two infected guys, then here we have 4 non-immunized persons. And in this schema, only this one was unfortunately infected by the disease. But these 3 blue guys were protected by the effect of cocooning. Cocooning means here you can see the cocoon of silkworm, or 日本語 in Japanese 蚕の繭ですね、これね。This non-immunized guy was protected by the wall made by immunized people. Wall made by immunized people protected this unvaccinated guys. Okay, this is cocooning effect. So, and this leads to herd immunity. And in previous 2 studies, Pfizer and Oxford vaccines showed cocooning effect. It means they can make herd immunity, even if not all people get vaccinated by these vaccines. Still, the community itself can get herd immunity, and the community can be preventable from the COVID. This is the great thing. So here we can conclude that messenger RNA vaccines and the viral vector vaccines, at least Oxford vaccines, truly prevent infection. Congratulations. This is the summary of efficacy or effectiveness of these vaccines. Next, we should switch to the dark side of the vaccines, side effects or adverse events. First, I touched on what is reactogenicity. Reactogenicity is defined as here, subset of reactions that occur soon after vaccinations and the physical manifestation of the inflammatory response to vaccination. This is very natural reaction. because each vaccines, not only in COVID vaccines, but also for all of vaccines in the world, contains some kind of pathogens or pathogen mimickers. So human body naturally react against those pathogens or pathogen mimickers. So human body, what human body makes against those vaccines is very natural. And we show pain, redness, swelling, ingression in the site of vaccination, or we may show fever, myalgia, arthralgia, headache, blah, 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 blah. These are all very natural and anticipated ones. We call these symptoms after vaccinations as reactogenicity. And as you know, COVID vaccines, especially messenger RNA vaccines, shows a high frequency of reactogenicity. And some people who got messenger RNA vaccines still do not show these kind of reactogenicity. And some of my friend doctors or friend nurses, hey, I didn't get any fever or any arthralgia, any pain. Did my vaccines still worked? No worry. Even if you have not get any reactions, any reactogenicity, still your vaccine is definitely working, no worry, okay? Many of you may know each of COVID vaccines show various kind of reactogenicity in high frequency, such like this, and but all of them spontaneously relieved within a few days. And this reactogenicity is more frequently showed in the second shot. And in Japan, Moderna vaccines has been approved, and they has begun to utilize from several days ago in the mass vaccination places. And so we should know about Moderna arm. We have two messenger RNA vaccines from Pfizer and Moderna, but almost only Moderna vaccine shows these kind of delayed local hypersensitivity reactions. It is, as you see here, erythematous, relatively demarcated, pinky to red, large scale of macule. And these macules are shown on the site of injection. But this is same patient. These 4 pictures are all from one same patient. This patient had a large macule on his injected site, as well as other pinky or red rash on his elbow. This is Moderna arm. And these skin reactions appears 7 to 8 days after of each dose. So it is after acute general reactogenicity. But all of them spontaneously resolve in median 3 to 6 days with or without treatment. If they feel some itchiness or pain, of course, we should give them pain reliever or some kind of a topical steroid or something like that. But they will be spontaneously relieved in few days. And this is never contraindications for the second shot, even if they get this reaction after first shot. This is totally has no relation with anaphylaxis. So please encourage the person who get Moderna arm after the first shot to get second shot, okay? But some patients again have Moderna arm after second shots. Yeah. And of course, we have been experiencing anaphylaxis due to COVID vaccines. And its frequency is shown as here. Pfizer, in case of Pfizer, 4.7 in 1 million vaccinations. And Moderna, in case of Moderna, 2.5 in 1 million vaccinations. And in case of inactivated influenza vaccines, we have 1.3 anaphylaxis in 1 million flu shots. So it's a bit higher than influenza vaccines, but I think we cannot feel in reality the higher incidence of anaphylaxis compared to influenza vaccines because it is very, very low figure. And potential allergen is thought that polyethylene glycol I mentioned before, PEG, and Oxford vaccines instance is not well published, but I guess this is also near the same figure. And Oxford vaccines contains potential allergen of polysorbate, and you may know polysorbate and polyethylene glycol shows cross reaction against anaphylaxis. So theoretically, if one person get anaphylaxis by messenger RNA vaccines containing PEG, then the person may also have anaphylaxis against Oxford vaccine. So theoretically. So this is what we should be careful. And from these reports, younger females are much more prone to anaphylaxis due to messenger RNA vaccines. And one hypothesis is because polyethylene glycol is frequently contained in cosmetics. And many females use cosmetics in their daily life. So it may be the reason, but not established. So if we vaccinate to younger females, we should be more careful to the acute reaction after vaccination. And here we move to the unexpected, more severe form adverse events. But before we talk about the real reactions, I request you to clearly distinguish 3 factors: adverse events, side effects, and coincidence. Adverse events are all ones reported by physicians, and all reported events are going to be registered in the adverse events database of each government. In case of Japan, Ministry of Health, Welfare and Labor continuously register all reported events after vaccination on their database and continue to get open those data on via the internet. And in case of Japan, a special advisory board for vaccine adverse events continuously assess these adverse events in terms of causation. And if such advisory board define that some of these events is caused by the vaccine, then it can be said side effects. It means causation, yes. But if those advisory board denied the causation of adverse events and vaccination, then they should be called coincidence. It means no causation. So if you look into such kind of registered database of adverse events after vaccinations, you should be careful how advisory board decided these events. But here we have very severe form of thrombotic events after vaccination of viral vector vaccines, including Oxford ones and Johnson & Johnson one from US. Now here we have 4 independent studies, 4 independent case series of thrombotic events. And each of case series contains such kind of cases, and its mortality is quite high. It around like a 30 to 40% case fatality rate. And almost all cases, almost all patients showed cerebral venous sinus thrombosis, CVST, and subsequently they showed severe thrombocytopenia. Platelet count was ranged from 10,000 to 12,000. And also, almost all patients showed positivity of antibody against platelet factor 4 heparin complex. And the etiology between this kind of special antibody and these viral vector vaccines is not still established. And the specialist is continuing to investigate the determining the etiology. One hypothesis is it may be a protein contained in these viral vector vaccines, or the other hypothesis is it is maybe due to vector virus itself, but they are not still established. And the incidence of these event, this is named vaccine-induced thrombotic thrombocytopenia, VITT. This event, VITT incidence, is around 1 to 4 per 100,000 vaccinations. And here, the incidence of CVST in general population is such a figure. So it may be higher than general population. And clearly, almost all patients has this kind of special antibody. So the relationship or causation of these viral vector vaccines to VITT may be definitive, may be. So this is a big problem, I think. Then we may have next question such like this. Then do messenger RNA vaccines have such an unexpected severe adverse events? And in these half a year, in these 6 months, 2 diseases, one is ITP, the other is Bell palsy, were suggested that it may be likely occurred after messenger RNA vaccinations. But I investigated these 2 studies, and both of studies are not, or both of articles concluded that no associations were seen so far from messenger RNA vaccines. And then one more thing we should think is mRNA vaccines so far have been shot at least for 500 million people worldwide, or it may hit to 1 billion in Japanese, 10億人ですよ。So, messenger RNA vaccines have been utilized for at least 500 million people in the world. Then any extremely rare undiscovered adverse events would arise hereafter. So here we can calculate such a thing, probability that adverse events of very rare, 1 in 10 million, never occurs among after 500 million shots. It can be calculated in such a formula, and the result is here. 1.393 multiplied by 10 raised by power of minus 22. It's almost zero. It's almost zero. So after 500 million shots, still rare adverse events is hidden. After 500 million shots, the possibility of hidden rare adverse events is extremely low, almost zero. So we can say messenger RNA vaccines would have no unexpected adverse events so far and hereafter, okay? But we should still be careful. If any unexpected adverse events in specific subpopulations would be uncovered and would be discovered hereafter. So here you see, I just knew this information just 4 hours before my talk, this talk, and I couldn't investigate any further details, but US CDC announced on 27th May so just 2 or 3 weeks ago, that since this April, reports of myocarditis and pericarditis after messenger RNA vaccines in those older than 16 years old are increasing in their database named Bayers. And still CDC is gathering further information and has not determined the association between messenger RNA vaccines and reports of these myocarditis or pericarditis. So still there is a possibility of just coincidence, not causation of myocarditis and pericarditis after messenger RNA vaccines, but we should keep eyes on this information. Such a thing may happen for specific subpopulations, even on messenger RNA vaccines. And in terms of adverse events, here I introduce our JPCA, our Academy's new proposal to of the safest point of jab. Now we are running time, so I do not talk details about this, but basically in a long history for deltoid muscle injections is have been done on the 3 fingers below the acromion around here. But after investigations, various kinds of articles, and we concluded this is not the safest. Here is the safest. So if you are interested in this issue, please refer to YouTube, but this is only Japanese, sorry. Or you can see each of our cited articles here in the list. In reality, I myself searched more than 35 articles regarding the safer point of deltoid muscle injections, and And also we appreciate great thanks to Dr. Nakanishi in Nara Medical University. He is an orthopedist and who has advocated for the first time in Japan to this issue, and he kindly supervised us, our academy, to make the previous video. So this is the great work from Dr. Nakanishi. And this is almost last part my talk, constellations for specific subpopulations. And we have such a kind of subpopulations: pregnant women, breastfeeding women, children, oldest old, previously COVID-infected persons, or immunocompromised or cancer patients. And as much as I know, for these 4 categories of subpopulations have been shown that they can get safely injected by COVID vaccines. And these many kind of articles showed its grounds. But still, for immunocompromised patients or cancer-having patients, have potential for some kind of unexpected adverse events or low effectiveness against the vaccinations. So here, if we are consulted by such kind of subpopulations, we should make dedicated, detailed communications with those people. And for example, for pregnant or breastfeeding women, I recommend to make such a communication. Do not easily link any pregnancy-related events after your shots, and or have you enough communicate with each of stakeholders, like your partner, your husband, your parents, or his parents? Didn't you decided to get vaccinated only by yourself? Such kind of detailed communications is needed, I think. And for immunocompromised or cancer-having patients, balancing matters. Balancing is very important between potential high COVID mortality of such kind of patients and potential low vaccine effectiveness and potential worsening of the disease itself by vaccination. And potential unknown adverse effects may happen. So balancing of such kind of issues is very, very important. And we should, if we are in primary care settings, we should encourage those patients to communicate and discuss with doctors in charge of the such disease. Okay. So these were today's my talk, and this is my last part. This is just a free discussion time. Let's discuss whether vaccination will make us free or not. Many people are now interested in whether they remove their face mask after vaccination or whether they can go out to drink with friends or families after vaccinations. What do you think of such issues? From my point of view, at least we healthcare professionals to keep strict restriction even after vaccinations. It is of course due to medical point of view. It means even vaccinated people still have chance to get COVID, and as well as from social point of view, we were prioritized to get vaccinated because we are healthcare professionals. And if we healthcare professionals to make us free, beforehand other general people, then those general people may get angry. Why you healthcare professionals do such a thing? So at least we healthcare professionals still keep restrictions even after vaccinations. But also we should think other things like if we get close contact with COVID patient, still should we vaccinated healthcare professionals do like 2 weeks self-quarantine? Or could we go out to our job still after the close contacts to COVID? This is a very important issue, and we should think of it. And each hospitals or each facilities may decide whether to do or not for after vaccinations.
Yes, I agree with that point. I think probably each facility has different human resources and different environments, so they have to discuss in the facility and in country as well. In Japan, we have no priority if you have vaccinated, but in the US or in Israel, there are some countries they have priority for vaccinated people. So I think it's very depends on the situation.
Yes, and one more thing we should think that it's like vaccinated people may be allowed to visit hospitals for inpatient hospitalized families.
Mm.
Still, they are restricted to visit their beloved family, hospitalized family in hospital. But if both of them have been vaccinated, they may be allowed to visit hospital or visit elderly facilities. We should start to think also about these things, I think.
Yes, exactly.
Yeah, I agree. Important points. Thank you, Aki. It's a brilliant presentation, really clear overview of COVID vaccines. Anyone else have any questions? Please.
Maria Sensei.
Hey, hello.
Hi. Thank you for your lecture. And I'm a little bit surprised that the Oxford vaccine, effectiveness of the Oxford vaccine dropped down suddenly after 3 months of the doses. And there are also some concern about immune to the viral vector virus itself.
Yeah.
And maybe I'm not sure if there is any data but is there any data which shows that the second dose of the Oxford vaccine can effective or not? After 3 months or 4 months, is there any trial going estimating the effectiveness of the revaccination of the Oxford vaccine?
Mm-hmm. Thank you for your very important questions. And for your first questions about immunity against vector virus, I have not searched in terms of such an issue so far, so I have no answers against. And of course, it is theoretically concerned. And you may know clinical phase 3 trials of Oxford vaccines have very interesting story of the fail of determining the first dose. And so as you know, in the phase 3 of Oxford vaccines, lower dose in the first shot and standard dose in the second shot. These arms showed more higher efficacy than the standard dose and the standard dose arm. So from this result, immunity against vector virus itself had some effect on the total efficacy in the different arms. So it is their theoretical concern. And a latter part of your question, I'm not sure about any clinical evidences for the second shot more than 3 months after effectiveness. And maybe UK government is continuing investigation in terms of this. And their mass vaccination campaign has just 6 months history. So I guess the effectiveness after the second shot of more than 3 months interval will be shown in the near future from UK. So we will keep our eyes on those results from UK government.
Mm-hmm. Thank you. Yeah. And can I have another question?
Yep.
According to the WHO, there is a DNA, about 10 DNA vaccine is on the clinical trials, but you didn't mention about that. So is there any reason?
No, I don't have any specific reason. I'm just interested in the approved vaccines and their real world data. Okay. Yeah, but I should check the other under investigation vaccines also. Thank you.
Okay, thank you.
Thank you very much, Aki Sensei. I have no question but comment. I asked you before that one of my friends, and she's breastfeeding her baby, and she are thinking if she get vaccinated or not, because she is a healthcare worker. She works as a healthcare worker, and we can just provide her this is a fact and this is evidence, and she should talk with her family and communicate with others, and then she can decide. And as the target generation gets younger in Japan, we medical doctors are faced to such kind of situation more frequently. So we have to think about how to describe and how to promote the vaccines. And I think it was very important to communicate with patients.
Yeah. Thank you for your very important comments. And as you mentioned, we may encounter such a dilemma in the very, very near future, like in weeks or in months. And to effectively communicate with such people, we need at least to obtain much information and much detailed and accurate information regarding COVID vaccines. And I'd like to keep my activity to distribute such knowledge for healthcare professionals, including medical doctors in Japan. Thank you.
Yes, thank you. I try to forward your information. Okay, thank you very much, Aki-san. So if there is no question from the classroom or online, this time is late and we'll finish this lecture kindly given by Aki-san. Thank you very much.
Thank you very much.
Thank you very much.
Very much.
Thank you.
Thank you.
Thank you
very much.